Hello, everyone, and thank you for joining us for this webinar series of which is co-sponsored by Gut Microbiota for Health and Ferring Pharmaceutical. And we're going to be today talking about the future perspectives in Clostridium difficile management, and also we're going to talk about the microbiome therapeutics. We have two amazing speakers, Ben Wallish from London, United Kingdom, and Ken Lunn from United States, and I'm going to introduce them to you in a bit more detail later. But just to again, just say a big thank you to Ferring Pharmaceutical for supporting this webinar series, which has generated quite a lot of interest. This is the final one in the series, in the current series, and we hope that we'll be hosting a few more of these in the future as well. And going on to the housekeeping, just announcements for our attendees. So you will be seeing a control panel on your desktop, which looks a bit like what you see on the screen, and you will see there that, and if you're using a mobile phone, this will look a little bit different, and you will see a question and answer icon there, and if you click on that, you will see another screen which allows you to type in your questions. You will not be able to ask questions directly, but you have to type in all your questions in this box, and then these will be visible to me, and then I can ask the questions at the end of each presentation. You will also see a chat icon there, and if you and you can use that to just tell us about any issues that you're experiencing during the session. If your video gets frozen or you can't hear us, you can let us know that. And if our panelists decide to use a poll, then you will see a poll come up, and you just have to click on the right answer, and that will become then visible to us. So really, without further ado, I'm going to request, first of all, Ben Wallish, who's a NIHR academic clinical lecturer in the Department of Metabolism, Digestion and Reproduction at Imperial College London. He's a post-CCT fellow in gastroenterology and hepatology at St. Mary's Hospital. He completed his PhD at Imperial College London and has previously been an NIHR-funded academic clinical fellow. So over to you, Ben, who's going to be talking about the clinical view on C. difficile infection and management in the future.
Thanks so much for the invite and giving me the opportunity to speak to you today in this exciting webinar. So keen attendees at previous webinars will find the background and epidemiology of C. difficile very familiar to them, but just to give a little bit of clinical context to some of the discussion that we're going to get onto today. C. difficile still presents a considerable clinical burden, an enormous clinical burden globally, and latest figures showing approximately 500,000 cases per year in the USA. Furthermore, the archetypal cohort of an elderly frail patient is not necessarily the archetype anymore. This evolving epidemiology and the growth of community cases, often young and healthy people, sometimes with minimal exposure to antimicrobials or even none at all. And the burden of C. difficile has a number of important broader clinical implications. Morbidity and mortality is pretty obvious, which is more than 50% in the most advanced cases of disease with toxic megacolon and multi-organ disease. There's a huge impact on the healthcare resource and health economic implications for healthcare systems. And impairment of quality of life is just as important, with this data from a few years ago in the bottom right showing how impaired this is for the patients that we look after. Many clinicians here will be very familiar with the progression, the challenges from a microbiology perspective of C. difficile infection, particularly over the past couple of decades. The emergence of hypervirulent strains, particularly those listed there, particularly 027, has created a huge burden on an even worse burden on healthcare systems. There are high rates of non-response to typical first-line agents, such as metronidazole, and there are high rates of recurrence, as listed there, up to 65% after the third CDI episode. Here are the latest guidelines summarized there from ECMID, published very recently over the past year or so. And as you can see, a variety of different choices are given at different aspects of the patient pathway. Why is that? Well, there's a number of different reasons, but part of the reason relates to potential issues with current gold standard of care therapies. For daxamycin and bezilotoxumab in particular, there are aspects related to cost. For daxamycin and bezilotoxumab have a number of other drawbacks as listed there. And regarding FMT, there is still, despite the explosion of interest regarding FMT, there are still question marks regarding regulation, and there are still concerns regarding availability. This is work from Simon Baumvor and colleagues over on the bottom right of this slide, which gives a summary of FMT use throughout Europe as a snapshot. And you can see the huge disparities in FMT use that do not directly relate to burden of disease within those countries. So we've still got some way to go. So back to my title, what does clinical management of C. difficile infection look like in the future? Well, I want to tackle this from a number of different approaches, but one thing I want to say is perhaps we don't necessarily need to reinvent the wheel. And perhaps firstly, we need to see if we can use the existing interventions we've got somewhat better. As an example of that, this is a study from 2018 from Guerri and colleagues, the EXTEND study, looking at rather than using just a standard fidaxomicin 200 milligrams twice daily for 10-day regimen, what would be the impact of an extended pulsed fidaxomicin regimen? A randomized study including 364 patients, at least 60 years, for a huge number of different European hospitals, where patients were randomized to receive conventional vancomycin dosing or an extended pulse fidaxomicin regimen as listed there. So drawing your attention particularly to the fact that this can extend up to 25 days. Patients were stratified by a number of key clinical variables. If you look at the demographic table here, a preponderance of women, approximately a third of cases severe. So looking pretty familiar to the sort of cases we might see in many of our typical clinical practice. And the primary outcome was sustained clinical cure at 30 days after treatment. Okay, so here we go. On the left, just to navigate you here, on the left-hand side of this chart, here is clinical response. Numerically, some superiority in clinical response to vancomycin, but statistically no different from the pulsed fidaxomicin. However, fidaxomicin really wins the day when you look at the other key clinical metrics. So in terms of recurrence, rates of recurrence with vancomycin are much higher than with pulsed fidaxomicin, and similarly, sustained clinical cure is much higher with fidaxomicin. So fidaxomicin winning out on all the key metrics that we would look at frequently. There's also interestingly looking at gut microbiota and particularly looking at the Shannon index as a measure of alpha diversity. And the diversity metrics favor fidaxomicin. In other words, fidaxomicin, as many of us will understand already, seems to be more protective towards the gut microbiota. It doesn't have the sort of broad disruption of microbiota diversity that's associated with vancomycin, which is a reflection of its specificity. There were a small and comparable number of adverse effects in both arms. What about rifaximin? So rifaximin, certainly from the UK perspective, is licensed for travelers diarrhea. We use it in hepatic encephalopathy, of course, but of course its attraction is this is a GI-specific, non-absorbable antibiotic that is well tolerated, few side effects, etc. This was a study from Giles Major and colleagues in Nottingham, a randomized study published a year or two ago in Gut, where patients who'd already completed standard of care anti-CDI therapy were randomized to receive either fidaxomicin or placebo for up to 1 month follow-up, with the primary endpoint of recurrence by 12 weeks. And over here is the Kaplan-Meier curve summarizing results. So over on the y-axis here, proportion with relapse, time from randomization on the x-axis. And there are obviously with placebo higher rates of relapse than with fidaxomicin, even from early time points. A significant reduction in recurrence risk in rifaximin with a total study size of 151. So this is something that we know is a C. diff therapy. We don't often think of it as a sort of first line or a major go-to C. difficile therapy, but this maybe means that agents we've got already are things that we should be thinking about even more. What about new drugs? To go to something more exciting. Well, firstly, when you talk about new drugs, you have to say the graveyard of drugs that there have been over the past few years, with a number of pharmaceutical agents looking promising at early phase studies, but failing to meet endpoints at later phase studies, including those listed there. However, one that's looked more promising over the past few years has been this ridinilazole, a small molecule confined to the GI tract. So all the factors here that look very attractive: very high inhibitory activity specific for C. difficile in vitro, effective in a hamster model, and doing very well in phase 1 and the phase 2 study. The phase 2 study is summarized here, where ridinilazole outperforms fidaxomicin in terms of lower recurrence and sustained clinical response. But the phase 3 results for this, which have recently been reported in abstract form, look more complicated. Here's a study that was reported in abstract form at ID Week earlier this year. So the phase 3 recruited 759 patients, randomized to ridinilazole or standard of care vancomycin. And here are the results. Sustained clinical response, 73% in the ridinilazole arm versus 70.7 in vancomycin. Clinical response recurrence rates as shown here. So what are the things to bore out? So even though in terms of primary response rate, there doesn't seem to be any difference between ridinilazole and vancomycin, there are some interesting secondary endpoints where ridinilazole may appear to have the edge, including recurrence rate 8.1% in ridinilazole versus 17.3 in vancomycin. You can see more about the time course and the dynamics for that over on the bottom right here. And ridinilazole also associated with preservation of gut microbiome diversity, preservation of secondary bile acids, of which more later, and fewer antibiotic resistance genes in patients' gut microbiome. So a difficult scenario about where ridinilazole yet. Definitely scope for hope, ongoing discussions with the FDA from what I understand, but failing to meet its primary endpoint in a very large study. What about CDI prophylaxis? This is something that the clinicians on the call will be asked about a lot, and it's an area where it's sometimes difficult to know what the right response to give. To go back to those recent ECMIC guidelines, only a very limited good practice recommendation is given, saying that it's appropriate only in very particular circumstances. To take some of the agents that we know well, a Cochrane review concluded probiotics may be effective if baseline CDI risk is greater than 5%. However, most recent clinical studies suggest only approximately 1% risk. So is this clinically relevant? Do we actually see patients like that? Retrospective studies have suggested anti-CDI antibiotics may reduce the risk of future CDI by up to 30%. There are some small RCTs, some small prospective studies with some contradictory results. But what we have to think about is if we're giving anti-CDI therapies as a prevention for C. difficile, what's the impact of the gut microbiome? What might be the off-target effects? And could this be, for instance, increase the risk of VRE if we're using vancomycin? This is an interesting study in which 100 patients who'd recently been hospitalized and received antibiotics were randomized to receive vancomycin or to placebo for up to 3 months. And there was seen to be a reduction in C. difficile in those receiving vancomycin. However, there's been some question marks raised about this study because it was a very high rate of C. difficile onset, 16% in patients receiving placebo. So this is definitely not an issue where we've got full resolution. What are the other strategies that may be emerging for CDI prophylaxis? Indeed, there's some new agents here. Ribaxamase is an orally administered beta-lactamase. And the idea of this is that when we're giving IV beta-lactams to patients, that some of that may spill over into the gut, harm the gut microbiome, and increase propensity to C. difficile, another sequelae of gut microbiome disturbance. So this was a double-blind, multi-center, phase 2 RCT, recruiting patients hospitalized with pneumonia who were treated with ceftriaxone for at least 5 days, who received ribaxamase or placebo for during and for up to 72 hours treatment with ceftriaxone, with the primary endpoint being incidence of CDI. Some of the demographics of the included patients are shown here, and on to the results. And what a result it is, whereby a p-value of 0.045 using a one-sided p-value, statistical test, showed there was a reduction in rates of C. difficile reduction, 2.4% absolute reduction. To break that down specifically, out of the 206 patients in the placebo arm, there were 7 cases of C. difficile infection, In the 206 patients in the ribaxamase arm, there were 2 cases, 3.4% versus 1%. So ribaxamase is still in the game very much after this study, and further studies are planned. What about a different strategy? So rather than an enzyme to break down beta-lactams, what about the idea of an absorbent agent that might be able to absorb antibiotic that spills over into the gut? This is DAV132, a colon-targeted adsorbent agent that uses a charcoal-based agent. This has been used in a recent randomized study, 243 patients, whereby patients hospitalized requiring a prolonged course of fluoroquinolones were randomized to receive DAV132 or placebo. And DAV132 use was resulted with very good absorbance of fluoroquinolones, as demonstrated by a 98% reduction in fecal fluoroquinolones, more preserved gut microbiota diversity, and then on a further degree of experiments, increased ex vivo resistance to C. difficile colonization. So mopping up fluoroquinolones from the guts, perhaps not surprisingly, makes the patient have a degree of resistance and resilience against C. difficile infection. So beta-lactamases, ribaxamase, and DAV132 absorbent agents are 2 interesting strategies that definitely show promise in terms of prophylaxis against C. difficile and may have a broader role overall. What about vaccines? And many people that followed the C. difficile vaccine story will have seen a number of highs and lows. Unfortunately, it's been outweighed by lows rather than the highs. There's been some further breaking news in this sphere this year. One leading recent candidate from Sanofi, which was a toxoid vaccine, got to a phase 3 study, but this study was terminated at interim analysis due to apparent futility. And you can see on the Kaplan-Meier curves the enormous number of patients that had to be recruited into this study before, unfortunately, it was clear that this was futile. And my understanding is development of this vaccine has been paused. Pfizer have also reported some results from a phase 3 study, their Clover study of their vaccine candidate. And from the data that's been available, what I understand is this did not reach the primary endpoint for prevention of primary CDI, but did meet some secondary endpoints, including a key secondary endpoint for reduction in medically attended CDI, in other words, complex CDI that required physician input, and reduced duration of CDI episodes when they're recurring. So the Pfizer candidate may still have promise and may still have some skin in the game. So while this is a sort of mixed bag for prophylaxis, and we're ending on a bit of a sad note here, I think there are sunnier horizons when we start to talk about what are the alternative approaches other than just using what we've got better, some new drugs or new strategies for prophylaxis. Now, Ken is the expert on microbiome therapeutics, and I won't say too much about that today, but just to give a little bit of a teaser, perhaps an introduction ahead of his. One of the sort of key papers that's come out in the FMT arena in C. diff this year has been this Lancet GastroHep paper from Simon Baumvoll and colleagues in Aarhus in Denmark, a small randomized study where patients with a primary CDI or a first recurrent CDI were randomized 1-to-1 FMT to placebo of a capsulized product after they had completed several days of vancomycin, standard of care therapy, including vancomycin. And this trial was stopped early on at an interim analysis, given that it was thought unethical to continue because those in the FMT arm were given such a reduced recurrence in C. difficile recurrence. So while we conventionally several years ago, we often heard descriptions of FMT being end of the line and experimental and only to be used as a last resort, this is perhaps paving the way for microbiome therapeutics to be used at an earlier point. This has also been noteworthy for a next generation FMT product being on the horizon and doing very well in a clinical trial. So this was a randomized clinical trial of a product from Series Therapeutics, CL109, based around Firmicutes spores, which was given as a capsulized product versus vancomycin. And in this study, CL109, versus placebo in this study. And there was a very clear advantage to the patients taking CL109 versus taking a placebo. So again, opening the possibility that we can move on from FMT with all its drawbacks to a more refined next generation product. And Ken will definitely have some reflections on this as well. Recognizing the impact of FMT and microbiome therapeutics, the topic comes up about Can we exploit research into FMT and what this shows us about colonization or gut microbiome host interactions or mechanisms of efficacy of FMT, and use this to make even more novel targeted microbiome therapeutics? To give you an example of what I might mean by this, this was research that I was involved in during my PhD, whereby we looked at bile metabolizing enzymes and the dynamics of those in patients with recurrent C. difficile treated with FMT. And we saw that bile salt hydrolases, these key enzyme group that removes the taurine and glycine group from primary conjugated bile acids, increase after FMT. That's over on the top right here. And that taurocolic acid, one of the key taurine conjugated bile acids, decreased after FMT. On a sort of comparable note, in that series SEER-109 study, it was seen that secondary bile acids increased within the gut very rapidly after FMT and remained at a high level for up to 8 weeks of follow-up. And that you can see that similarity between that and the dynamics for engraftment of the species in their product as well. So what can we start to do? Can we exploit FMT research? And this is a nice review from Vincent Young and a colleague recently, who's saying that the emerging fields of microbiome research into FMT, microbiome host interactions, and C. difficile beyond is ripe for exploitation and for as a source of potential novel therapeutics, microbiome therapeutics. Knowing what we know about the role of bile acids within the gut and their impact upon the germination and vegetative capacity of C. difficile, maybe we'll get to a stage where we can give specific bile metabolizing enzymes or even specific bile acid metabolites themselves, rather than conventional either as an adjunct or alternative to conventional agents. We also know that FMT has some effect through its impact on short-chain fatty acids. We also know gut microbiome properties, including antimicrobial peptides or the bacteriophages within the microbiome as well, may be key to the efficacy of FMT. And it may be that we can exploit this knowledge as well towards novel microbiome therapeutics. What other strategies might we exploit in future as well? Well, the whole C. difficile life cycle and the whole environment in which C. difficile lives are all ripe for exploitation. And here are some of the examples of what we might be able to do. So I already mentioned this idea of bacteriophages. It may be that we can get viruses that live within our gut microbiome and exploit those as a novel targeted therapeutic in an anti-C. difficile fashion. It may be we want to exploit something in the C. difficile environment, including the biofilms in which it lives, aspects related to our ionic transporters, target epithelial damage and inflammation, target VEGF-A. So there are a whole raft of approaches that we might exploit that are at some at earlier, some at later phases of research and development into potential therapeutic agents. So to bring all these themes together, what do I think the future is looking like? I don't know exactly the direction of travel, but I think there's lots of possibilities. And overall, I think the future is looking bright. We can repurpose or we can use existing agents smarter. We have novel antibiotics on the horizon. We have a range of different possible prophylactic strategies, some with novel mechanisms of action, including the ribaxamase story, including charcoal absorbent agents. In terms of microbiome therapeutics, we can use what we have potentially earlier. FMT is a good example of that. Or there's the arrival of next generation FMT products and eventually even potentially more refined, more reductionist microbiome therapeutics. And our increased understanding of biology of CDI and of microbiome host interactions more generally allows even further avenues for exploration. Thanks again for listening. Thanks for giving me the opportunity to talk, and happy to share in the debate any questions. Thank you.
Ben, thank you very much. That was a truly brilliant talk. Really enjoyed it, and very comprehensive indeed. We have about 5 minutes or so for questions for Ben, and of course at the end of the session we can also take further questions. But if anyone has any burning questions now, please go ahead. Please type in your questions in the Q&A panel that's on your screens. But perhaps Ben, if I may just ask, it's very clear that some of the newer antibiotics are very much more effective than the traditional ones, although there may be some cost implications, but they will be offset by better treatment and less recurrence. But in terms of the best therapeutic options that are available for recurrence, you showed that very interesting data on FMT at first recurrence, and then an excellent response to that. What is the current thinking around that? Is about clinically, what is the best approach for recurrence? And of course, that may depend on the age of the patient and the risk factors and so on. And is there a group of patients where you would go for FMT straight away, and another group where you would actually go for some of the traditional?
Yes, so that's interesting. So when we wrote the BSG HIS guidelines a few years ago, after a lot of discussion among the guideline development group and among our stakeholder consultation, we came to a position where we said, look, conventional pharmaceutical agents are very well established. We have a very clear dose. We have a lot of experience with them, and so we said earlier use of FMT would be associated with people with conventional risk factors associated with higher risk of recurrence. That might be where you want to use it earlier in the disease course. But I think the emergence of studies whereby it is being used earlier, coupled with the time that has gone on and our experience of FMT and lots of those earlier concerns that we had about safety signals, either not being such a concern, or us knowing how we might mitigate those risks in terms of aspects such as donor screening, have meant that there is increasing confidence of safely using FMT earlier in the course. So I think it's very difficult to give a one-size-fits-all answer to that. When we use FMT still, we have a multidisciplinary team discussion to decide about what our alternative strategies are, what our exit strategy is if the first one doesn't work. But I suspect even that's going to look quite antiquated quite shortly. Yeah.
Okay, thank you for that. A few questions that have come through. Does fiber-rich plant foods intake decrease prevalence of C. diff because fiber-rich diet offers rich microbiota?
Yeah, I think I don't 100% know the answer to that. When I've looked at these studies before, and it's a bit like the answer to probiotic studies, the difficulty with this is there is heterogeneity in design, there's heterogeneity in the supplement that's used, there's heterogeneity in the patient group, the studies are generally small. So I don't think there is anyone who would say, that that's a major, a clear therapeutic option, although it may be something where there is a rationale to recommend it. So I deliberately didn't put anything specific in about that, and I think it's interesting. And given the biology of what we know about fiber, it obviously seems something prudent to look more into. But I don't think we're at a stage where we can give anything conclusive of clinical pertinence.
Thank you very much. Is there any evidence of antibiotic resistance to rifaximin?
I can't remember what was mentioned in the study that I mentioned there. I certainly don't remember anything about it. It's certainly not something that can particularly think of as hand. But that's not to say that it hasn't been an issue. It's not something I can think of as being a major issue offhand.
And final one, which is in some countries FMT is a drug, in others it isn't. So what do you think is the best option here? What would be best to make it more useful and more used in clinical setting?
I'm going to kick that one to Ken, because I think that he is, with his experience, he is someone who has really grappled with this, and clearly the way to go is to get something that is, one of the major issues that regulators have with this is when we started using FMT, it was something that isn't regulated, where we don't have a dose, where we can't say that every unit is reproducible, etc. And that's one of the key issues for regulators, and that obviously regulators would feel much more comfortable with that sort of approach to a microbiome therapeutic product. So I think Ken probably has some more nuanced reflections on that than I might do.
Ben, thank you very much indeed. Really enjoyed your talk. Thank you. And we move on to our next speaker, which is Ken Blount, who's the Chief Scientific Officer at Rebiotics, which is a company from Ferring. He's also the Vice President for Microbiome Research at Ferring and is a scientific expert on a number of topics, including antibiotic discovery and development, fecal microbiome analysis, and RNA targeting therapeutics. And today he's going to talk to us about future perspective of microbiome therapeutics. So over to you, Ken.
Thank you. Good. I will assume you can see my deck. Thank you, Dr. Mullish, for the really nice background and setup. I think it flows very nicely into what we're doing at Ferring Pharmaceuticals to develop new microbiota-based approaches. I've been in this field for some time, fighting against C. diff in the antibiotic arena. I've seen many of the things that you had presented, particularly the antibiotic compounds. And I think as I was developing those, FMT, or fecal transplant, was beginning to come of age and become more commonly used. And I always, at that time, 10 years or more ago, said, you know, someone should really develop a potentially approvable therapeutic that's harnessing that FMT. It turns out that was happening around the same time I was saying that, and it was a company called Rebiotics in Minnesota in the USA, and that was founded by Lee Jones in 2011 with the guiding philosophy of can we take the promise of FMT and convert it into a standardizable drug product. And this is sort of what we just talked about in that last question, was the heterogeneity of FMT among sites. How do you prepare it? How do you store it? How much is in it? How do you administer it to patients? None of that was really standardized. So the goal from the beginning of Rebiotics with the founder and CEO Lee Jones was really to do that and to transition it to an approvals process. And in the FDA, that was clear from the beginning that that would be through the investigational new drug process, or an IND. Now, it's not clear exactly throughout the world if that will be regulated the same way. We can touch on that briefly, but in the USA, that was, we started engaging with the FDA in 2012, and they gave the first
guidance. I can't see your
guidance.
slides yet. I don't know whether you've actually started.
You don't see my slides?
No. Can you do a screen share?
I think I was, but I'll double check. Thank you for letting me know. There.
Yeah, that's fine. If we can.
Let me change the
Yeah,
view.
fantastic. We can.
Good.
Yeah, that's better. All right.
Very good. So I apologize for that. So this was sort of a timeline that I like to use of what we were thinking at the beginning and some of the key bullet points along the way. One of our guiding philosophies was not only to make a potentially approvable drug product, but to do so in a way that would provide a patient-centric view. So that meant things like accessibility, what is the minimal patient impact required to provide this to patients. So a lot of that went into our thinking from the beginning: connection with patient advocates, connection with clinicians. That was very important to our DNA from the get-go. We first interacted with the FDA in 2012. That's when they issued their first guidance on live biotherapeutics. The key question posed at that time was fecal transplant. Is it a transplant or is it a drug? The FDA was rather quick to say it's a drug. It's not human tissue. It's a drug. Now, today we might argue or debate or consider it differently as we're appreciating the microbiome as a new organ. But it's very clear in the FDA that these types of microbiota-based live biotherapeutics will be regulated as drugs. Around that time in 2013, there was also a guidance issued that was, in essence, a loophole to that IND guideline. So the IND guideline says any microbiota therapeutic has to be developed and run clinical trials within the FDA guidelines for IND. Enforcement discretion said, we choose to take the discretion not to enforce this for fecal transplant for C. diff infections. Why did they do that? They issued a guidance and then they said, but there's an exception to the rule. They did it because patients needed it. Frankly and very clearly, patients needed an option. There was a recognition that a microbiota approach could provide an additional benefit to patient, but there was not an approved option, and therefore, prudently, the FDA said we need enforcement discretion. And that's continued in some fashion to this day. But again, our first engagements were back in 2018. Our first clinical trial started in 2013 with an investigational product we call RBX2660. And that's defined here. It's a microbiota-based live biotherapeutic. That means what it sounds like. It's based on microbiota. It's alive. It's regulated as a live biotherapeutic, or LBP. It's standardized. We've developed it to be stable to storage over long term. And there's a quality control program in place that is closely aligned with the FDA and monitored by the FDA. A version of that is shown here. It is a liquid suspension. It's prepackaged, 150 milliliters total. And it did have the designations I showed you here. And it's administered rectally without any, none of our clinical trials required bowel prep. It was administered rectally. So we've run over the course of the last almost 10 years, we've run a large number of trials summarized here. This started with our phase 2 trial, which was aimed at feasibility and safety and what is the patient experience, what is the clinician experience. We moved into our first and the industry's first randomized placebo-controlled trial. We added a large open label trial that was really aimed at safety and efficacy and additional patient experience. And that led to a pivotal phase 3. And I'll get into these in a little bit more detail, but I think an overview is important. That was really aimed at demonstrating substantial evidence of effectiveness. In parallel, we started a large open label phase 3 shortly thereafter. And this was aimed at more of a real world patient population. Many times when we do clinical trials in pharma that are directed at an approval, we have to be scientists. And that means that we have a controlled population and perhaps narrowly defined so that confounders to our interpretation are minimized. What that can mean, though, is that there's a fair amount of patients that don't fit those narrow criteria required for science. And that's a challenge, first and foremost, because patients could potentially benefit from access. It's also a challenge in that traditionally, as an industry, we recognize that sometimes an approved drug may come to the market without being fully evaluated in a typical population that a clinician like Dr. Mullish would encounter. This trial, this phase 3 open label, was really aimed at broadening those enrollment criteria to include comorbidities that might normally be excluded. So for instance, IBD, IBS, those were included in this trial, really an aim of getting at what is the real world population and what is the safety and efficacy in that population. And then there was a retrospective study. At one point, for quite a long time, we additionally allowed RBX2660 to be administered under that enforcement discretion policy. And then we worked with several investigators to collect data from that enforcement discretion. So it's a large clinical trial program. All of our controlled trials were designed in the way that I'll show you on this slide. When we encountered a patient that would be candidate for enrollment, they would be undergoing treatment for an active recurrent CDI episode with an antibiotic. If they completed that antibiotic successfully with a resolution of their C. diff symptoms, then they could go on to their assigned study treatment in our trial. If they did not resolve their C. diff infection, they wouldn't have received an additional treatment. And that really is summed up in our title. The clinical trial was aimed at reducing recurrent CDI, not treating active CDI. That's very important. Once that resolution was complete, there was a short washout period after which an assigned study treatment would be administered, either RBX2660 or placebo. and or placebo, depending on the trial. The treatment success was defined as absence of CDI diarrhea for 8 weeks after study treatment. And then every trial had an extended follow-up period for safety and sustained clinical response. In parallel, we collected stool samples both prior to treatment or baseline and at several time points after treatment. And the goal there was to monitor the microbiome and the bile acid composition, really to assess what were the changes and to start to understand how this was working. Dr. Mullish presented to you some data showing the importance of those. This has always been an aspect of our clinical trials program throughout. So just to get back into these trials, way back in 2013, we completed that first trial. And that was really focused again on safety and feasibility. So questions like, what is the clinician experience for administering this rectally? What was the patient experience for administering this rectally? Did it work? It was the first type of investigational product of its kind that had been manufactured through GMP processes. The short answer was the safety and the efficacy were highly consistent with the FMT literature. That fueled us then to go to our next trial, which was this phase 2 trial called PUNCH-CD2. It was our first randomized double-blind placebo-controlled trial. One of the key questions we sought to ask in that trial was 1 versus 2 doses, as well as versus placebo. And the reason we asked that 1 versus 2 was you see a lot of the FMT literature. If a patient initially fails to respond to treatment, then they can get another FMT, or they have been administered another FMT treatment. Very often that second one will then resolve and prevent the CDI. So we saw a similar outcome from our first trial. So in this second trial, we asked the question, what if we just plan to give everyone 2 doses 7 days apart? Does it increase the benefit? Short answer, no. There was no added benefit of the 2 doses versus the 1 dose of RBX 2660. So you'll see later on, our phase 3 trial then used a single administration of RBX 2660. The trial that we ran in 2015 was a larger open label safety trial. Again, it was aimed at what is the long-term safety, and this included a 24-month follow-up, which is quite a long follow-up for most clinical trials. But given that this is a new modality, altering potentially the microbiome, we in the FDA felt it was really important to assess long-term safety. What we observed was it was well tolerated, mostly mild to moderate adverse events. 79% of participants experienced that initial treatment success of being CDI diarrhea free at 8 weeks after. Of those, 91% sustained that CDI free status to 24 months. So long-term durability observed. And then again, that leveraged into our phase 3 trial, in which we showed that RBX 2660 was superior to placebo. This was a single administration. This was also an additional patient population. All the trials prior to this included only multi-recurrent patients. That means they had to have at least 2 CDI episodes prior to the enrolling episode, so a total of 3 episodes. This case was at least 1 prior, so now 2 total episodes, including the enrolling episode. And we did observe treatment success after 1 administration superior to placebo. We also saw that in a 6-month follow-up period, 90% of those primary responders remained CDI free through the 6 months of follow-up. And then the safety was consistent with our prior trials in that only mostly mild to moderate adverse events were observed. We also observed that the patients, the microbiome composition and diversity were significantly shifted. I'll talk to you about that on the next slide, as well as the fecal bile acid compositions. So specifically, what I'm showing you here is a lot of complex data. What you want to look at is on the y-axis, you're looking at the relative abundance of specific classes of bacteria. So for instance, 0.2 relative abundance means 20% of the total bacteria present in the stool was a certain composition. So what you should see from that is I've highlighted 4 classes. In red are the relative abundances of participants in that phase 3 trial at baseline or prior to our assigned study treatment. You can see that in red, Bacteroideia and Clostridia bacteria classes are quite low. Compare that to a composite of healthy, where those are generally the majority of a healthy microbiome. Very different. We also saw at baseline in this trial, gamma proteobacteria and bacilli were elevated. Many gamma proteobacteria are pathogens. There were some patients that had 50% Klebsiella at baseline. That's not what I would characterize as a healthier microbiome. But as you can see here, within a week after the study treatment, patients begin to resolve from that baseline towards the healthy population, with increasing Bacteroideia, Clostridia, and decreasing gamma proteobacteria and bacilli. So this was evidence to us that significant changes were observed from before to after treatment, and that they were shifting towards what we would consider a healthier population. We did see significant differences between RBX 2660 treated and placebo treated, and it's very important because we're assessing. We know that after a patient receives an antibiotic, if they never saw our clinical trial, you can expect some level of restoration. But what we asked with the active versus placebo was how much different or potentially better is it when they received RBX 2660. In short, we saw quite significantly higher restoration or increase in Bacteroideia in the RBX treated compared to placebo. We also looked at bile acid compositions. Now, I want to go into a little bit more detail on this. As Dr. Mulish introduced, these are rather important, both to the microbiota as well as to C. diff. Our liver will produce primary bile acids. Those are only converted to secondary bile acids by healthy microbiota. Our body doesn't do that conversion on its own. So if we have a healthy microbiota, generally we'll see mostly secondary bile acids in the stool. If we don't have a healthy microbiota, you'll see a predominance of primary bile acids. That's important to C. diff because some of those primary bile acids actually promote C. diff to germinate and to grow, whereas some of the secondaries will repress C. diff from growing. So they're intimately connected to both healthy microbiota as well as the C. diff life cycle. As you can see on the right, what I've done is I've plotted for you here the ratio of the secondary bile acids to the primary. At baseline, all of our participants had a very low secondary to primary bile acid ratio. That means that the predominant bile acids were those primary, consistent with the microbiota data I just showed you that they didn't have a healthy microbiota. We saw then a shift as early as 7 days after, which then continued throughout that 8-week treatment and beyond, but not shown here, of an increase in that ratio. So it shifted over to over 40-to-1 ratio of secondary to primary. That's highly consistent with the microbiome, and it's highly consistent with the prediction that you've shifted to a state of the microbiota or the bile acids that's more resistant to C. diff colonization. Importantly, we did see, as I showed you here, that the RBX responders had a higher shift compared to placebo responders, which not surprisingly was higher also than any of the treatment failures. We went back now and collected all of the data from multiple trials, and we presented this last year at DDW. This is just summarizing that we saw a very similar restoration or shift in the microbiome and the bile acid among 3 different clinical trials. This includes hundreds of patients now. We're seeing a consistent shift, most notably gamma proteobacteria and bacilli predominated before, and Clostridium and Bacteroideia predominated after, and then a shift from primary to secondary bile acid predominance. So all of these were consistent with the model and the prediction that we set out with, which was that a restoration could be achieved. I do want to emphasize that this is all exploratory analyses. I think the field as a whole is still trying to get to a true definition or description of mechanism of action. And I also want to emphasize that the FDA views it that way. I think the FDA very clearly feels that the world and the field are still in an exploratory phase for fully understanding this mechanism. But this was very promising data consistent with the model. So more recently, we looked deeper at that microbiome change, and we went beyond saying, here's what happened. What we saw, and I showed you, was restoration. And that's really saying, here's what changed from before to after treatment, and here's how those changes were different when I intervened with an active versus placebo. The next phase is when we saw a shift after treatment, did those shifted bacteria, did the newly appearing bacteria come from my administered live biotherapeutic or somewhere else in the environment? And that's what I would call engraftment, is incorporation of the bacteria that were administered as part of the treatment. I'm going to go through this really quickly, but we just presented a new set of data last week at a conference, really addressing that question. And you really have to go to extremely deep sequencing methods and some novel bioinformatics to ask that question. And what we observed was that a large number of bacteria were in fact engrafted directly from RBX 2660 into the participants in our phase 3 trial. This is a total number of different species that were observed to engraft in the treatment group, the RBX treatment group, at 1 week, and then it continued and was maintained out to 6 months. As a control analysis only, we shifted our bioinformatics to say, does our method indicate engraftment in placebo-treated patients? And as you can see here, we found 0. Now, it's an interesting control to do because the question you're asking is, did I get bacteria from my treatment into a patient? I think the field is still grappling with what some of these drug development terms mean, but the predominant view that's emerging is that engrafted means a bacteria came from a treatment and took residence in a participant. So technically, it's not even a logical question to ask with a placebo, but we felt it was important to do that control. And you see the result here is no engraftment observed with our method in placebo, whereas significant engraftment in the actively treated patients. So this, again, is sort of building that story. I think as a field, we're getting there. We're definitely getting there, but there's still a long way to go before we can claim any of this is true mechanism. So really, this summarizes the clinical data and the supporting microbiome and bile acid data I've just shown you. Many of you probably are aware that we recently had an advisory committee with the FDA, which was quite an interesting experience, but it's really just signifying that this industry is getting to the point where we're starting to have late stage discussions as an industry with the FDA about could there be an approvable product. We've also looked over the course of that time. One of the questions posed to me in this webinar preparation was what about beyond CDI? In short, I think the potential is clearly there. We've run a number of trials, either ourselves or as investigator sponsored, looking at other indications where the microbiome is thought to be important. And that's really summarized here. Most pointedly, we did see a signal of efficacy in a pediatric ulcerative colitis trial. The reference is shown here. So I think, in my opinion, I think the future is really bright. Just to summarize where we are now, this has changed so much. In 11 years since the first regulatory guidance came out from the FDA, we've seen a significant maturation of the guidance and process in the US. We've also seen other regions progressing. We've been discussing with EMA and other regulatory agencies in Europe and around the world how these would be regulated. And I think I've seen tremendous progress too. So we're hopeful that the industry as a whole will be poised for the first LVP approvals. And that really, even now and in the future, we expect a tremendous diversity of modalities, mechanisms, clinical trials. And I think it's really has a potential as a field to be transformative to the patients. So with that, I'll thank you and take questions.
Okay, so that's brilliant. Ken, thank you very much. That's very interesting, novel data that you've showed us. We have one or two questions coming through, which I'll start with, and just a reminder to everyone to please type in your questions on the Q&A panel. So Gerard Clark, one of our colleagues from the ESNM, has asked, do you see any benefits for accompanying dietary guidelines to support engraftment?
Sorry, what was the question?
So would you see any benefits for any accompanying dietary guidelines? So would diet have an effect to support engraftment?
Yeah, that's a good question. I think it's a question that hasn't been answered. As Dr. Mulish indicated, a lot of the dietary studies are very heterogeneous, and the guidelines for understanding precisely how those affect microbiome are still developing. Intuitively, we will probably say at some point in the next 20 years, we start to see clear direction for dietary connections to microbiome, but I don't think we're quite there yet.
Okay. And may I just ask, obviously this is still classified information, but the restoration that you saw in the stool samples after treatment, is that consistent with what was expected?
Yeah, that's a good question, and it's not confidential. We've published a lot of this work from multiple clinical trials. It was very consistent with what we expected. So I think what we showed you was a shift towards what's considered to be a range of healthy individuals. There is no gold standard healthy, so it's a range, but we certainly saw a shift towards that range.
Yeah.
And into the range.
That's certainly an issue as to the huge variation that you have, inter-individual variation that you have.
Yeah.
That still within that, you see that signal that there is a change in the microbiome in a very specific pattern. So that I think is quite interesting, which I haven't seen that come through in other data related to probiotics or microbiome products.
Certainly not probiotics, but I think there is data with FMT and other investigational therapeutics really telling us that the whole modality has the potential to shift in this direction.
Yeah. And in terms of positioning this product, the RBX2660, do you think the positioning is going to be actually, as you've done the trials in recurrence patients with recurrent disease, or do you think actually there could be a possibility of actually preventative treatment as well?
Yeah, I think I really can't speak to whether it could be a preventative. All of our trials focused on the reduction of recurrence after a primary antibiotic in recurrent CDI patients. So whether or not it would shift to prevention after a preventative after primary antibiotic, really, we don't have the data to speak to that today.
Yeah.
I think, as Dr. Mulish pointed out, price is a consideration in this field. And, fidaxomicin, the taper data looks fantastic that you showed, but again, the challenge there is the cost of fidaxomicin compared to vancomycin.
Yeah.
So.
Okay. And I think in your as you were introducing your methodology and the type of patients that were included, you did mention irritable bowel syndrome, but it wasn't mentioned in your outcome slide. So is there any data on any improvement in symptoms in IBS?
So we didn't look specifically at endpoints that were IBS defined. We looked at CDI-directed endpoints with IBS patients included. So it's slightly different. So I wouldn't want to make any claim about IBS, but what we did see, at least an interim, was that CDI patients with a co-diagnosis of IBS fared just as well with resolving C. diff as patients without IBS.
Okay, all right, that's interesting, and I'm sure once the product is out there, then lots of other indications tend to crop up. So where exactly are you with the project at the moment? As to what is the sort of route that the company will be taking from?
So we remain and have always been aimed at taking this to the market as an approvable. I mentioned that we had just completed the advisory committee. So the next step beyond that is further discussions with the FDA about potential approval. I couldn't share timelines, but that's really where we are, is aiming for those later stages.
Right. Okay. Okay. That's really brings us to the to our time limit for this webinar, and it remains for me to just thank both Ben and Ken for their fantastic presentations. Really enjoyed it, and the quality of both the talks was amazing, very informative. So thank you to both of you, and again to Ferring Pharmaceuticals for sponsoring this webinar, and all our other colleagues who've been involved in this webinar series, including our administrative staff at ESNM, who've been providing us with a fantastic support. So thank you very much indeed. And, I'll close the webinar now.